I'm delighted to be here. My name is Mehrdad Haji Babai. I'm at the University of Guelph, near Toronto. And two of our co-chairs are here as well, Florian Lies and Christian Meisner. And this was an exciting session to organize. In fact, there were so many abstracts that the organizer decided to divide our session into 2 days. So half of the talks are today and half of them are tomorrow. And yeah, so it's about the use of environmental DNA in some sort of in a standardized fashion in actual environmental assessment and monitoring programs. And I'm very excited to hear the presenters. We have an excellent lineup. But I let also Florian to say a few words about the plans that we have for the session and going forward with this discussion.
Yeah, thanks, Mehrdad. Some brief words about the session. What we realize, we have great tools, but we want to see them implemented. And there's kind of a lag between this research that's being done and the implementation in routine monitoring programs. So how to bridge that gap? What are the necessary steps? We hear several talks. We have a couple of posters. What we invite you to, we have an open access journal running under Pensoft. That's the publisher. You probably know it's a big biodiversity publisher. So metabarcoding, metagenomics, I'm the editor-in-chief there. Mehrdad is also an editor. We have a couple of free papers for early career researchers. So if you would like to submit to this special issue on standardized DNA and eDNA-based assessments, let us know. We will also announce that tomorrow. And all those that are interested, just write us an email. We will then write about the next steps, how to proceed with that special issue. Yeah, with that, I guess we are ready to go, or I'm looking at the first speaker. So, yeah, very much looking forward now to introduce Donald Baird as the first speaker this morning. I hope we get his presentation. Yeah, well, almost. So the first presentation has the title, Connecting Biodiversity to Biomonitoring, Applying Environmental DNA Metabarcoding in Large-Scale Bioassessment Programs. Donald, as soon as something appears on the screen, the stage is yours. And if someone wants information on the journal, by the way, we also published an eDNA guideline from DNAquanet, the European Cost Action. You can find that upstairs at the Pensoft table, and there's information on the journal. What the mission? Submission date. We have to find a deadline, probably latest in half a year from now, but the point is every time an article is ready, it will appear, and it's collected under the special issue. So we have to agree on a final submission date, but I would not be too restrictive. But I would like to have really a focused special issue, how to bridge the science implementation gap. Now I see that the presentation is up. Donald.
Should be ready to go, eh?
Yes. I don't know. Guess we can. No, that's back. Yeah.
Okay. Okay. Good morning, everybody. Thank you for all coming to this session to hear my presentation. What I'm going to try and do in basically 10 minutes this morning is to give you some introduction to a large project we've been involved in, which has been going on since as we call it, the before times, before the pandemic. The project has proved to be much more complicated than we anticipated, and we're still working on it, but I'm going to show you some preliminary results this morning, and also really try to begin to address this question about how we can get on-the-ground biodiversity information at scale. Here we go. So the background to this is, if you look at the paper on the left-hand side, Wendy Monk, a colleague, and I were asked by Canadian Council of Ministers on the Environment to do a report back in 2000, I think it was 2009, on the status and trends in biodiversity in Canada's lakes and rivers. We were quite surprised when our aim with this report was really to try to look at national scale consistent evidence for trends in biodiversity in fresh water. We figured we might have struggled to get data for many different groups of organisms, but we thought things like fish and so on, we'd be okay. In fact, what we found was a general lack of consistently, sorry, spelling error there, consistently observed biodiversity data at national scale. This was then followed up by WWF Canada, who carried out a Healthy Waters watersheds project, where they were trying to look at threats and status of Canada's watersheds. They came to the same conclusion. In fact, we were kind of involved in providing input to that project, and we did let them know about that at the beginning. And I can say even today, we still have a problem that in terms of biodiversity, biological data, Canada's watersheds, in terms of large-scale, consistently observed data, are data deficient. So the question really here is, oh good, I see all my slides are going to be cut off at the bottom. So can biomonitoring data provide a useful biodiversity signal? Why am I interested in this? Well, we have a national biomonitoring program in Canada. It collects consistently observed data across the country. Canada, obviously, at more than 9 million square kilometers, with a relatively small population for that land area, is a very challenging place to try to do this type of work. But we figured that where there is a biomonitoring program, particularly one that's based on a network of networks, there's the opportunity there to perhaps use data within that program, or to help move that program towards collecting the type of data where we can look at status and trends in freshwater biodiversity. So what I'm going to do here is now I'm going to look at a comparison study that we did between data that's collected using the normal national program methodology and another project that I think Merdad and Terry Porter will be talking about later in the session this morning, the STREAM project, which looks at using DNA metabarcoding as an alternative approach. Just to go through quickly, we looked at wadable streams and rivers, running water habitats, 3-minute traveling kick sample, which is a multi-habitat sample, so it's a good integrator of freshwater macroinvertebrate biodiversity. That's the target group we're looking at. Those are commonly used internationally for river monitoring. Where it diverges is with microscopy, we're looking at a 300-count subsample as our way of assessing composition, and in this case, relative abundance within the sample. And then we also did DNA extraction of whole sample homogenate using replicated PCR with multiple target sequences. So we're not just using one primer, we're using multiple primers in order to get a more complete and overlapping information. In terms of the type of data that are generated, this was one of the problems with the national program as it is at the moment. The level of taxonomic resolution is relatively coarse at order or family level, but we are looking at single sample count data for relative abundance. Whereas with DNA, we can go right down to OTU or ESVs, but we chose for this project to look at genus level ID based on a 99% sequence match. I'm not going to go into the details of how the sequencing and bioinformatics was done, but we can provide input in that later if needed. And we looked at primer-specific presence-absence. So here, again, keeping with the same kind of parallel stream of data, we had a range of sample sites. We didn't look at the same samples, but we looked at samples that were collected at the same time. So we collected a duplicate samples. So, and also there was good overlap between the two data sets, but not complete. So there is, but basically they're targeting the Canadian maritime provinces of New Brunswick, where I'm based at the Canadian Rivers Institute, Nova Scotia, and Prince Edward Island. And this gives you an idea of how many reference sites and test sites we looked at. Reference sites are based on a subjective expert judgment criteria. Test sites are sites that are believed to be potentially impacted. One of the challenges we've had, and this is from some work we published previously, when you look at comparing direct observational information relative to DNA-based biodiversity signal, they don't match. There's always going to be mismatches, and there are many different reasons for that. But ideally, I mean, this diagram here kind of looks at the relationship between what's observed in a microscopy-based sample relative to what you get with DNA. And ideally, what you want is all of those bubbles there to be in the top right-hand corner. And as you can see, that's not the case. So already we see, these things are not the same, but they are also probing the environment consistently. To give you an idea, this is just average taxon richness across these samples. What we're looking at, I mean, we're looking at a large number of organisms, types that we're observing, but in terms of per sample richness, obviously with the DNA, we're getting greater taxon richness relative to the traditional methods, so there's no surprise there, really. But we were quite surprised with the initial preliminary analysis. Before we really did anything, we thought we'd do a quick non-metric multidimensional scaling plot to look at the overlap between reference and test sites, because obviously that's the primary purpose of biomonitoring, is to be able to discriminate these things. And we were quite surprised to see really a remarkable separation between reference and test sites using DNA, which was really not mirrored with what we got with the typical microscopy-based sample analysis. Now, if you notice there, the stress score on the right-hand diagram is pretty high, but it was a large sample size. And we are still investigating why this is, and I'll touch on that in a moment. Basically, it comes down to one of the big differences between biomonitoring sample processing and DNA metabarcoding of extracted benthic samples is that subsampling is employed for the visual identification, because it's just not practical to process every specimen in the sample. So right away, we know that these two methods are probing the system with different levels of effort, if you like, across the whole sample. Now, I know that certainly with DNA, we are taking aliquots from a homogenized sample, so we are kind of subsampling as well. But remember, this is homogenized DNA, so the probability of missing things, and we've looked at this using replicate PCR, is pretty low. So what's the consequence of this? Well, let's look to see within the national program, when people traditionally do biomonitoring work, how much of the sample do they actually observe? This is a multi-habitat sample. How much of it is actually observed and identified? And the answer is not really very much. If you look at, this is based on the national program of 3,500 sites. This looks at all the major kind of drainage areas across the country. The main thing I want you to look at there is the kind of median value across all these box plots. You can see it's less than 25% for most cases. In some cases, 10%. So therefore, you're collecting a sample and you're really only looking at a very small part of it. So we think perhaps part of the reason why we get better discrimination is because the DNA is giving you a more complete picture of what's in the sample. Why is that important? Well, on all other things being equal, the less common taxa within the system, when you constrain the number of organisms you look at, particularly where there's things that are overabundant, you're more likely to miss those using that type of method. So just to summarize, DNA metabarcoding, what we found, can detect more macroinvertebrate taxa more consistently than traditional sorting methods and ID methods. Microscopy coupled with fixed count subsampling detects taxa haphazardly, leading to more false negative detections. Consistent detection using environmental DNA provides increased confidence in separation of reference and potentially impacted sites. And existing biomonitoring methods that use fixed count subsampling for example, our national program could be failing to detect adverse change through type 2 error due to an inability to consistently observe less common taxa. Where we're going with this now is to look at developing a new biomonitoring approach based on multi-species occupancy modeling, based on primer-specific detection and occurrence probabilities for observed taxa. The data then will have the dual purpose of biodiversity discovery and also trend reporting, and also can be used for site-level bioassessment. And that's the link I'd like to make to the presentation that's coming later this morning. The diagrams on the right-hand side look at what we've been looking at in terms of species occurrence and inherent detectability using different primers. And again, I can discuss that if there's interest. So again, I'll just thank you for, I'm just over by 4 seconds, I think, here. So just to thank all the people who've been involved in this huge project, particularly the community-based monitoring, including indigenous communities that have helped support collection of samples, and all of the funders that have helped support the work over the years. Thank you very much.
Thanks a lot, Donald. We have time for one quick question. May I? Oh, okay, there are microphones. Very good.
You're dealing with stream communities, right? So I'm imagining that the size class of the insects and the community overall, it's highly heterogeneous.
Oh, yeah.
Yeah, so I'm wondering, how do you normalize the size class when homogenizing these tissue samples to avoid the effect of biomass bias, maybe?
Well, we don't. The sample basically is for DNA extraction, the sample is cleaned to remove excess organic material, and then the whole sample is homogenized.
Okay, thanks once again. Further discussions in the coffee break or in the lunch break. We move to the next speaker, and I'm very happy we have Michio Kondo from Japan here. I recently was in Japan for the eDNA Society conference, and I can only tell you I was really amazed when I saw his presentation there. So welcome to present the Anemone project.
Okay, this mic is on. Okay. Thank you for giving me this great opportunity to introduce about the Japan's eDNA monitoring network, Anemone. Okay. As all of you already concerned, biodiversity is now rapidly declining, and mainly it is considered mainly due to human activities. And nature positive has become a very important international target. The question is how we can realize the sustainable usage of ecosystem. And to make it realized, of course, we need a scientific understanding of the nature. It is very critical, given that the sustainable usage is about somehow optimizing the way to utilize the ecosystem. Given that ecosystem is a very huge, complex, nonlinear, dynamical system, we need sufficient data that is good enough to overcome the uncertainty coming from the complexity of ecological systems. And eDNA is a very great solution to get such a good data. Oh, it doesn't work? Oh, okay. I totally forgot. Yeah, this is the one. Thank you for letting me know. Okay, so talk about it. Yes, good. And environmental DNA is a great solution to get such a good data. And Anemone is the system through which we can obtain efficiently the eDNA big data. It is, say, a network of institutes including the governmental and academic and also business sector institute, to make it realize in a very interesting way. And it was founded in 2019. The target organism of anemone is a fish in coastal ocean and freshwater originally. Originally means now we are expanding the target. And the routine monitorings are conducted at 77 institutions. Some are weekly and others seasonal, and something other institute monthly. And every survey is conducted following the standard protocol, which is provided by the eDNA Society. Now it's 4 years ago. Yeah, this is the manual we are providing through the eDNA Society. And the number of surveys done by the anemone monitoring is, of course, keep increasing year by year. And now we have more than 5,000 survey records covering the for Japan. Okay. Hey. Okay. Anemone is characterized by its efficient division of labor system. While people at anemone node, they do field works, such as sampling. The anemone hub, they do everything else, including logistics, wet analysis, and dry analysis. And this enables basically everyone, even non-professionals, to be involved in the project. And with an easy-to-start instruction manual and sampling kit, direct supervision by professionals is minimized. This is the sampling kit you may receive when you join the anemone monitoring. You have filters and gloves, manuals, and other usual tools for the usual eDNA survey. And you have even a video manual provided, which you can watch while you are doing sampling in the field. Okay. The hub, once you finish sampling and filtering, you will give your samples back to anemone hub, and they do wet and dry analysis. And after the analysis, they give all the data back to you. This interesting system allow the local communities making a significant contribution to anemone surveys. In one program, which is conducted by or supported by the Earthwatch Japan, local people, they choose their survey sites based on their curiosity. And in the last 3 years, more than 300 participants conducted eDNA samplings and detected 800 species, fish species, which is 1/5 of all the open data we are now providing. And what is nice about this project is that it is financially supported by a private company. So this is, maybe you can say this is a threefold collaboration between academia and the local community and also a private company. And eDNA is called, sometimes people say eDNA is cost effective, but it can be still challenging when the access to your sampling site is really hard to make, such as alpines and underground lakes. Yet it can be made possible if you work with nature enthusiasts. And this year we had a very exciting project where climbers and hikers, they offered helping eDNA sampling in a mountainous area. And with financial and material help from outdoor companies, we collected more than 90 samples that are now under analysis. Okay, another place that is hard to access is the open sea. It's really effort demanding. if you like to regularly take eDNA samples in the open sea. Yeah, this also can be made possible if you collaborate with a shipping company. Yeah? Anemone and the shipping company, NYK, Nihon Yusen Kaisha, are cooperating to conduct a routine survey using their shipping route. Okay, so Anemone, it enables us getting nice eDNA data. But the problem is how to use it, okay? To enhance the better practice of using Anemone data, we make those data open, fully open. AnemoneDB is a system to realize it. AnemoneDB is a dedicated database for anemone. It is under operation since last year. The monitoring data are stored and made available to public, and it's freely and publicly accessible, and there are no usage restrictions. I just like to show how it looks, okay? If you go to the AnemoneDB website, you see this map. And if you click, then it shows how many samples is included, are obtained in that area. And if you click it, and you get some visualization of the species composition of each sampling effort. And of course, it gives data in a machine-readable way. And it gives sample information, of course, experimental information, and the number of DNA copies in water, because we are using internal standard DNA when doing eDNA analysis, so we can quantify the eDNA amount in the water. And okay, oh, I totally forgot again. Okay, yeah. This is what you will see on the website. And you can do many things with the anemone data. So you may focus on spatial distribution of thousands of species. And if you put genus richness against latitude, you can see spatial biodiversity pattern and how it changes between seasons. And if you put genus richness against day, you get high temporary, high resolutional time series. And you can see something happening around 500 days. Maybe we need some scientific investigation to identify what is happening there. And like this, much-site frequent monitoring data can give many interesting scientific opportunities. And interesting scientific approach made possible with such a massive eDNA data is to construct the JSDM, a joint species distribution model. And according to ecological theory, a species occurrence or community composition is shaped by interaction between the environmental niches and the species response to those niches. Okay? Those two variables are very important. I'm not to get into details, but the JSDM allow you to back estimate the unobserved niche and species responses to those niche axes from a set of occurrence data. We developed a JSDM framework applicable to massive data and applied it to the 1,200 species at 500 sites. And we identified 5 niche axes and species responses, which will explain the species occurrence pattern along the Japanese coast. Okay, so now we have anemone monitoring network and anemone OpenDB. Yeah? Those two components taken together helps making monitoring system open to public. However, there remains a very big issue, which is, I think, shared by all of you. It is how to get money, to support this monitoring system. And the Anemone Consortium is a cooperative group of industry, local governments, and academia to construct the economical mechanisms that translates that good data into economical value. And it was established last year, and now we have more than 30 institutional members. We regularly have meetings, maybe 4 or 5 times months, and keep discussing and exchange ideas of how we can utilize the anemone and its data for nature positive business. Okay, so this is the last slide. I introduced the Japan's monitoring network, anemone. The point is that anemone is not just a monitoring network. It is the anemone system taking together the anemone monitoring network, and anemone OpenDB, and anemone consortium. And it works as an ecosystem to realize the cooperation between different stakeholders and making monitoring sustainable and the best use of eDNA techniques to establish the society in harmony with nature. Yes, that's it. Thank you.
Thank you very much, Michio. We have time for 1 or 2 questions. Dominic.
Yeah, thanks for the talk. This looks awesome, and it looks like you are much further than the Europeans are. But my question would be, so you said you have a standardized way of processing your samples in the laboratory, which is stated in these guidelines. And as we all know, there's still much improvement in our work, like technical improvement in our workflows. So how can you adjust for that? Will you be using the methods you developed in 2019 forever? Or do you have a way to adjust for technological improvements?
Okay, actually the standard manual coming from the eDNA Society that has been revised several times. And now we are doing the 3rd revision, in which we are going to include the more bioinformatical analysis in it. And of course, some technical update about the wet analysis is also going to be included.
There's another question, Alison?
Could you give us a couple of examples of how users are using this data? How they access it and what they do with it?
Actually, there are many ways. But okay, I have to say, using the full data, people using full data is almost restricted to the scientists. They are using this data. If you are looking at the more local community, they are more, how to say, it's my gut feeling, but they are more likely to utilize the more localized data. For example, some people are using our eDNA data to identify the impact of the aquaculture on the ocean system by comparing the different sampling. And also other people, they are trying to see, what ecological positive effect, the nice way of, I don't know how it is called in English, but woodland management. Yeah, do you see?
Restoration?
Restoration?
Yeah, it's more proper way to,
Yeah.
Yeah.
Very good. We have to move on.
Okay.
Thanks once
Thank you.
again. I really think that's a fantastic model, and we should discuss this model also, how it can be extended, I guess, to other regions. We come to the next talk. We realize when it comes to eDNA uptake in official monitoring programs, more and more formal networks form in different countries. It's usually national programs, and Finland also published, I guess, about a year ago, a national roadmap, and Tiina Laaman will present the Finnish national roadmap on eDNA. Tiina.
Okay, hello everyone. It's my pleasure to be here today to show our eDNA roadmap, which we published with our colleagues from Finnish Environment Institute and from Natural Research Institute of Finland. So, first of all, I would like to discuss a bit that why we do need this kind of national and international coordination of molecular monitoring methods implementation. As we all know, these methods, they have now developed fast and they have reached that kind of critical point that implementation in routine monitoring has started worldwide. But the field is fragmented with different organizations fast developing their own methodologies, which cause a risk of duplication of efforts and incompatibility of end results. And as a community, it's really important that we understand that if we wish not only to jump on the train, but also go stir it, now is the real time of act. So we decided to do this eDNA roadmap project, which was funded by Ministry of Environment in Finland. And the aim of that project was describe the national state of the art in molecular monitoring methods, and then identify the main possibilities and challenges and development needs. After that, we of course need medium and short-term aims where we are aiming to go, and then really provide a concrete action plan for the next 4 years. And as soon as we started this roadmap work, we noticed that international coordination and collaboration and also cross-organization interaction in practice, they are really important to really get this into action. So what we did an international literature review where we were checking all the research papers for last 5 years, and then we ranked those in using this kind of technology readiness level scale to see where we are now. And based on results from 70 papers, this is still ongoing work, but this based on the 70 scientific papers, we can see that most of the papers, basic technology is now already developed in the field, but only few cases are actually in routine implementation at the moment. And we can see that there are quite huge difference. You see that light blue color, freshwater, is the most development one, and then marine environments, but especially in terrestrial side, there are still more like a piloting ongoing activities. Then, with the kind help of eDNA Aquanet, we made an international survey and translated many languages and got total of 171 responses to our survey to see that what are actually those key limiting factors currently, which are like causing that we cannot take these molecular monitoring methods into full action. And based on the results, we can see that key limiting factors, they are funding, lack of expertise, method standards, reference libraries, and cost of the analysis. And I guess we all agree that. And we also looked at it in national level and international level. And especially in Finland, the lack of funding and lack of know-how or persons who know how to do this, they were the major reasons to not to take these molecular methods into action yet. So then we were sending a lot of emails in our institutions to see that where we are actually now, what we are doing. And based on those results, we see that national efforts, they are quite diverse, but they are scattered and they are still largely in the testing and piloting stage. basically all our institutions, research institutions, are tested or used at some point of molecular monitoring methods. And there are only few cases, game species such as the wolf and European and Canadian beaver, that is already in routine monitoring. But there are quite extensive metabarcoding pilots ongoing, for example, phytoplankton, zooplankton, benthic invertebrates, diatoms, and fungi. So there are a lot of activities ongoing. So then in our roadmap work, we made a vision that where we're aiming to go. And we see that internationally standardized molecular methods, they will work as a cornerstone of a routine monitoring. We see that molecular methods are really important part of the future environmental monitoring, but they cannot always replace the traditional methods. And especially during the transition phase, where we are using traditional methods and new molecular-based methods, we need really extensive and coordinated method development and standardization. So then I will talk a little bit more about standards. So standards for measuring biodiversity with molecular tools. Because these methods are so fast developing and almost ready for routine implementation, lack of standard is one of the major reasons for non-adoption of these methods. There are quite many national and international level methodological guidance, but it's quite scattered, and minimum method requirements are not universally accepted or agreed yet. And because there are many funding, like a smaller funding available, it's quite easy to go like a national level in this guidance development. And as we have already heard, there are actually quite many funded international projects also ongoing, but one really important thing is that we really exchange the information between those ongoing activities, so that we can reach that point that in the end we have those standards and data flows will be globally comparable. So what is happening now? There are quite many nice big efforts ongoing. So SEN standard for water quality sampling, capture and preservation of environmental DNA from water has been published already. And then there are efforts to establish these international standards through eDNA working group in ISO, led by my colleague Christian Meissner from Syke. And then there are efforts to engage the international eDNA community in standardization. So it's really important thing that we work together on this. So what we recommend, if you are a researcher or somehow involved in using molecular monitoring methods, please use always global method standards to reduce risk and ensure molecular method update for reliable biodiversity monitoring and assessment and data comparability. And we really need very quickly and inclusively mobilize relevant international stakeholders. And we need to identify a relevant and mandate entity to oversee novel method update and to set a list of needed standards. Then I will talk a little bit about our ongoing activities. So we published that eDNA roadmap a little bit over a year ago. And it has been so nice to see that after that there is quite many national and international research and development projects running. And there are many different kind of funding opportunities. For example, our Ministry of Environment in Finland launched a Biomon program, and there are now quite many eDNA-teamed projects running in that program. Then there is Academy of Finland, European Research Council, Horizon Europe, Biodiversity Plus. So it's really nice that there are this kind of funding. Since when we were doing that survey, we saw that lack of funding is one really key issue here. So it's really, really nice to see that at least it's changing slowly. Molecular monitoring methods are also a part of our national environmental monitoring strategy, and it's just started on September, so it's really nice that we can put it into our national level that way also. And then one of those projects funded by Biomon program is called eDNA Monitor, where we are testing, developing, and validating the DNA methods for assessment of ecological status of water bodies. And there are many organismal groups such as phytoplankton, zooplankton, macroinvertebrates, and diatoms. Then we have a new Academy of Finland funded project called Spore Life, where my colleague Vera Norras is leading that one, and we are combining eDNA and flow cytometry with integrated atmospheric modeling for bioaerosols and biodiversity monitoring. So this is a nice one. And then in Finnish Ecosystem Observatory FAIR project, led by my colleague Petteri Vihervaara, we are just about to launch that kind of online interaction platform where anyone who is interested to use eDNA or other molecular monitoring methods can search easily information, what are these methods, what I can do about this, and if I want to make some kind of survey, how I can do it and who to contact. So we are building this kind of portal now. Then there's a really nice Horizon Europe funded eDNA Aquaplant project, which just started. There's a 18 EU partner project, which is promoting synergies, harmonization, and interoperability between existing activities. So this is really nice one, and it's good that we have this kind of project in European level. And this one is led by European Marine Biological Research Center, and we at the Finnish Environment Institute are part of that project. And then in national level, one really important thing at the moment, and I guess international level as well, is the data management. How and where we should store those data. And in Finland, there is a development project called FinBIF, which is preparing for the management of DNA-based species observations. And currently, different options are on the table. Most probably, we will follow the GBIF extended Darwin Go format, but of course, we are always interested to hear how and what other countries are doing, and always open to further international collaboration. For that, I would like to thank the organizers to let me tell you about eDNA roadmap, and here you can see all other roadmap co-authors, which we thank also. Thank you.
Thank you very much, Tina, and especially for stressing the international importance of linking things. So because we all want to get some food at 1 o'clock, I propose we directly move on to the next speaker, which is also the session co-lead, Mehrdad Hajibabeh, who will present, I don't see the title slide at the moment, a model toolkit for standardized biomonitoring in freshwater streams. Training, community-based monitoring, DNA metabarcoding, bioinformatics. Mehrdad.
Well, thank you so much, and I'll try to be quick. Yeah, this is actually a talk that was supposed to be given by Terry Porter, but unfortunately, Terry could not join, and she sent me her slide, so I'm basically the mouthpiece. I'm trying to do a good job presenting here. But it's a project that I've been involved and I've coordinated and led, so hopefully I'll be able to get the message across. So, I mean, we have been here talking about biodiversity, and we all know about sort of the decline of species and what's happening with the different types of negative impacts on biodiversity. But we also need to remember that basically it's sort of linked to also key ecosystems and habitats, and freshwater is one of those vulnerable and a really important, I would say, ecosystems that is being impacted. Here, basically, you see a map of the WWF Canada produced this a few years ago to show the state of watersheds across Canada. And like the take-home message is that the biggest challenge is data deficiency. And you can see those gray sites that we don't have any data. And as Donald mentioned, the traditional approaches that we've been using, they're not as scalable. They're not also accurate, and we know that, but they're also not as scalable, especially for a country like Canada with so much fresh water. Now, I'm not going to go into the details because Donald mentioned that, and you probably know that benthic macroinvertebrates are one of the key bioindicators in freshwater systems to understand the health of the ecosystem. But the challenge has been sort of how to operationalize and scale these, to deliver the data. And then, it's one of those sort of interesting moments in my career where Donald came to me in 2007, working on the barcode of life and sort of developing, barcoding systems. He came as an environmental scientist, within the Agency Environment and Climate Change Canada, with the challenge that he had faced, in biomonitoring the freshwater. And that started a collaboration that goes all along, and basically we introduced the first meta-barcoding in freshwater in around 2008-9, and we published it in 2011. So basically this idea of basically moving from the conventional biomonitoring towards using technologies such as DNA, and obviously it can be augmented with other types of tools like AI, image analysis, remote sensing, and so on, is to transform this system of biomonitoring. And with the DNA, essentially, we are sort of changing the interface from, let's say, looking at specific organisms, like one investigator, one bug, one microscope, to what we can see in bulk. And that's the transformative nature of this, which we have heard and seen with the eDNA movement and all the studies that are going on. But there are also challenges and difficulties. So one of the difficulties that I faced is when we had a couple of these big projects with Genome Canada funding and so on, is that how are we going to obtain these samples? So our national agency, they don't have enough resources to go and sample for us. We can get more sequences and crank up the sequencing. We can get better computers to do data analysis. But how are we going to able to obtain these samples from these watersheds, especially in Canada, where we have all of these remote sites? So this was where we were approached by WWF Canada and Living Lakes Canada. And they proposed that we bring our DNA-based approaches to the community-based systems or community-based monitoring, which is, I realize, it's a very healthy and ongoing effort. A lot of communities would like to know what's happening in their watersheds and ecosystems. They have adopted some of the traditional approaches, like the cabin approach that Donald mentioned, but they also had the issue of, okay, we send these samples, where are we going to send them? To taxonomists? They're going to have to wait sometimes more than a year or a year and a half before receiving the results. So it was a very interesting merging of these two different somewhat, I would say, very, very different disciplines. Like you bring in this very sensitive sort of DNA-based approach and pair that up with the community-based monitoring. And I was skeptical to begin with, how are we going to make sure that we don't get contaminated samples and so on? But throughout this project that I'm going to describe in the past 5 years, I think that's been an excellent example how we can scale up the use of DNA-based tools by the use of basically essentially democratizing this field by basically bringing it to the hands of citizen science and community groups and so on. These are all the watershed participants, organizations that are actively participating in the program in the past 4 years, and we are now sort of expanding into US, in Japan, in Scotland, and in other parts of the world. These protocols are all available for anyone to use. The 4 modules that we basically established for this project are standardization, automation, modularization, and scalability. And the goal was to bring down the turnaround time and increase the throughput. I'm not going to go into the details of each of them, but then defining project modules and defining basically tasks for different groups was really important from the beginning. So this is the sort of those 6 modules that we have currently, from the outreach and recruitment of the communities to training and certification. Basically they go through a rigorous training and then they get certified for doing this. We built that on the backbone of the cabin training they already had, but we basically added the DNA sort of approach to it. And then last year we detached it from the cabin, and then we have now in a standalone eDNA metabarcoding sort of certification program for the community groups. Then it comes to sampling, and then lab processes, bioinformatics and data analysis, and finally results and dissemination. And in every aspect of this, we have enforced some sort of a standardization. As I mentioned, an important part was the training that I told you, but we also have developed a standard bioinformatics workflow here that you can see MetaWorks. Some of you may be familiar with. This is the work that Terry Porter developed based on the RDB classifier. Is a classifier that has various taxonomic groups covered, including macroinvertebrates, but also we have other groups. And then the other important part is these reports that you can see in the bottom here. The standard reports that we have generated for the project, we have produced over 100 of these reports. These are basically simple biodiversity reports for the communities. When they send the samples, like we want them to quickly get some information. Now, this can go off, obviously, different stakeholders will look at the data differently. Donald showed you some of the work that he does with the sample, in terms of the modeling, comparisons, like predictive approaches and so on. But when you are working with the community groups, you don't want to just go and say, hey, collect samples for me, and then I'm going to publish the papers and you can be acknowledged in the paper. These communities want to stay engaged. They are true partners of the project. That's why I think that having these type of reports are really important to keep them engaged and informed. And up there you will see a storyboard of all the sampling events that we have had, and these are all linked to the stream website, publicly available for everyone to see. And then there it comes to basically lab automation, which we published a paper a few years ago in Molecular Ecology arguing that metabarcoding and eDNA approaches are really good for automation of different parts of the workflow, so that I don't really see any problem of scaling this up to cover millions of samples even. And you saw in Paul's talk today that basically where this is going in terms of the coverage. I talked about the community participants and their reports. I would say we also have developed a data portal for the project that basically you can access actively all the samples that have been analyzed. You can look at it from a taxonomic angle, a spatial angle, and these are not very difficult to develop. These are not tools that require a lot of programming. In fact, I had a grad student, and over summer we joined forces, and basically we put this portal together. So if you have projects, obviously want to develop these type of things, they can be used for various purposes as well. And then some of the impact that comes, obviously, immediate impact for the communities is really important. I give you some examples here. So one of the First Nation groups, the Blueberry First Nations, are using, for example, this data to look at the effect of industrial activities on their sites. Some of them are keen to know specific organisms like invasive species or some of the endangered species, like whirling disease in Alberta, for example, has been an issue, and then we've been able to harvest that data specifically for the communities and channel that to them, and so on and so forth. So there's some really quick and immediate impact of this type of work for the communities, but I would say this is contributing to the larger scale biomonitoring across Canada, as Donald mentioned. Obviously, there is a lot of scientific publications, technical publications, or biological and ecological publications as well, and I'm not going to go over them. But some of the trends that we are seeing, like the type of data that we are seeing, spatially, temporally, in terms of the biodiversity, and then in different groups of organisms. We have now added also diatoms to this, so it's not just macroinvertebrates, but we have optimized the same kicknet sampling for the diatom sampling, so you get diatoms as well. And then here is a sort of a biotic index that we have modified using the DNA meta-barcode. So we're moving towards, you saw the map I showed you with the WWF, and we still have a lot of data deficiency. But the good thing about this is that this method is scalable. If we start using the community groups more effectively, I think that with a fraction of the cost and a much faster turnaround time, we can produce data that can be then used for monitoring in a timely fashion for the systems. With that, I thank all the collaborators and Terry for putting together the slides, and thanks for your attention.
Thanks, Merdat. Also, for time reasons, let's have the questions in the lunch break. We move to the last 12 minutes talk, which will be given by Pedro Beja from Portugal. We will move to Africa. He will speak of challenges and opportunities of standardized biodiversity eDNA surveys in remote African rivers, the case of Korobal River in West Africa. So, Pedro, the stage is yours.
Well, good afternoon, or good morning still. I'll be talking a bit about our work in Africa and the challenges we are facing there. We have heard in these days lots of challenges about monitoring, but then think again and multiply by a large number. So working in these environments is really complex, and we have tried to find ways to get most data on areas that are seldom, if ever, monitored. So I'm doing something wrong. Yeah, so in this kind of remote areas that are difficult to access, most of the work has been done, if ever done, with conventional techniques. And conventional techniques have proved to be relatively effective in some areas to detect endangered species with camera trapping, with fishing, and so on. And the problem is that they are not scalable. It's difficult to get there. And so the idea is whether eDNA can help us solve the problem. And we have tested this in several rivers. We are working in several rivers in Africa. But in this particular case, we are working on the Korobal River in Guinea-Bissau. This is one of the poorest countries on Earth. It's a very remote area where it's difficult to access. And we have tried to sample across the river. We have done not as many sites as we are in Canada, but these are sites that are difficult to access. They are physically demanding for people to get there, even dangerous in some cases. So when we get there, we need to try to get most information that is needed. And so, again, so what we've tried to do is to sample along the stream. We have done 11 sites in 2022, that's what I'm showing now, and another 13 in 2023. So this compared to the samples that have been shown here is very small. It's a very small sample size, but believe it's very hard to do it. And we have tried to collect large amounts of water, so we do with boats and we cross the river. We do transits for integrating the entire river, and we try to, at each site, to have 2 to 4 replicates with high throughput capsules with about 30 liters per one, per replicate. We also do a lot of PCR replicates to be sure to detect the species, and we do it with standardized procedures using pipelines that we can discuss later, but not with me because I'm not the right person to discuss these kinds of things. And we have tried to target vertebrates, fish, mussels, and eukaryotes. So the idea is, in the one end, to have a general biodiversity survey of the areas, but the main goal is to detect rare species, species that are threatened and that may be used in that country for, in one end, attract conservation funds because they want to show that it's an important country, but also to try to guide management. We have tried to have large coverages to be sure that we are detecting all the taxa in the samples, and in this case, of course, it shows that we were succeeded. But of course, we were very far from detecting all the species that are, or all the ESVs that are in the river. So these are asymptotic estimates of diversity, and we have from 1/3 to half the ESVs, and probably even less. But so that means that we really need to have many samples scattered along the watershed to get the diversity of the catchment. Of course, the discrimination, the ability of identifying the taxa that we get varies a lot across markers. Some of them because the markers are not, they never have resolution to go down to species, but in most cases because we don't have the reference collections to understand what species are there. For instance, this is a good example. So it's a relatively species-poor group, the freshwater mussels, where at the beginning we had 47 ESVs and just 2 species identified, so lots of diversity that we didn't know what it was, but because there was a mussel specialist with us, he was able to collect the species, and then after barcoding, it was resolved in 8 different species, including one species that is critically endangered, and it was thought extinct in most of this river and only occurred in a very small area. For vertebrates, most of the reeds belong to fish, but we were able also to detect mammals and birds and other taxa. And we have tried again at the same time that we did the eDNA to collect specimens to build the reference collections. So we collect, when possible, the fish. We get the tissues and photographed and try to start building the reference collection. And then there are starting to be problems because, again, most of the species cannot be identified, but even the primers may not be the best for this kind of fish that exist here. For instance, you see there that with telio, so in principle, a primer designed for fish, we get less fish than in the bycatch of a general vertebrate primer. So this means that probably for this kind of environments, we need to fine-tune what kind of primers or what we are using, or using a wider battery of primers. But even with these problems, we can start to resolve different spatial differences in species distribution. This is, again, these are very preliminary analysis because we are still waiting for the results of the rest of the samples. But for instance, here we were able to discriminate the lakes, the headwaters, the main river. Again, this is just a first exercise, just for fun, to see that it works. More disturbing was that it didn't work very well with amphibians and reptiles, maybe because it was the wrong time of the year in the case of amphibians. Part of the amphibians were detected with a bycatch of the bivalves primers and with a telio primer. So again, probably the primers are not the best. And another surprise is that very, very few reptiles were detected, including no detection at all of crocodiles. Crocodiles were everywhere, but they were never detected. We could detect 2 turtles, including 1 critically endangered soft-shell turtle, but no crocodiles at all. It's something that we have repeated in Namibia with river full of crocodiles and no DNA from crocodiles for some reason. Again, birds, most birds were detected were from aquatic birds, but we were also able to detect species new to the country that were never detected there, maybe including, for instance, this rail that is a secretive species, and it showed up first in the DNA, and then people were able to find it. And mammals, that's another a good finding. We found that most reeds were from hippopotamus, so hippopotamus consumed a lot of the reeds from our sequencing, but we were able to detect many other species, including monkeys. Monkeys were very well detected by eDNA, whereas other species were not so detected. We detected for the first time in the country this giant forest squirrel that had never been detected before, and we detected endangered species that were not known to be in the country, or they were very rare, including this red colobus. And when this was presented this summer in Rwanda in the Conservation Biology Congress, there was a lot of people interested on this red colobus, which is a very rare species that occurs only scarcely in West Africa. We did an exercise comparing eDNA with camera trapping. It was not a perfect study because it was not exactly in the same sites, and it didn't not cover exactly the same places. So it's a preliminary analysis, but it shows some interesting trends. So of course, DNA was very good to detect aquatic species, and what I compare there is detected only with eDNA, detected only with camera trapping, and detected by both. And so the aquatic species were much better detected by eDNA, as one could expect. But then it was also better to detect arboreal species for some reason. It's understandable why camera trapping does not detect very well arboreal species, because the cameras are down and the mammals are up, but probably because they defecate in the water, or they for some reason, they were very good at detecting monkeys. Others were detected very well by both eDNA and camera trapping, such as, for instance, chimps were detected very well by both, or bushbucks. And then there were others that were never detected by eDNA, such as, for instance, aardvarks and animals that are probably rare and never or rarely go to water. So summarizing very quickly, so for these particular case studies, there is huge fish diversity, but we don't even know what species are there. So maybe half the species or 1/3 of the species have never been described, or certainly not barcoded, but probably not even described. We could detect species of conservation concern, from mussels to chimpanzees. So it's very good that we can detect this. We were able to detect species that were difficult to detect by other methods. But we also learned that some of the interpretation of the work, of the results, benefit from conventional sampling. So we could learn a lot, because there was also some conventional sampling that could let us know that we were missing some obvious things that were there, but we were not getting. So, and this will be the beginning for estimating community metrics. In terms of challenges, and eventually for discussing later, of course, these areas need barcodes, they need reference libraries, otherwise the value of these surveys it's good, but it's suboptimal. We need better estimate of errors, biases, detectability, because we can go there, we get eDNA, we get species, but how do they represent what is in the field is not known. So if we don't associate the detection with a detection probability of some kind, it's very difficult to have reliability of when we're showing. And for that, there's a lot of trade-offs that we still don't know how to deal with. Should we filter more water, or filter more filters with less water, and then to estimate detectability? Should we do more sites or more replicates per site? Should we do more technical or more biological replicates? So, and this is very important, because we are talking about countries where money is not, it's very, very limited. Projects for going there is very, very limited. So we need to have cost-effective methods that are reliable and allow us to say something with, at low cost, because otherwise it will be impossible. And again, complementing with traditional, conventional surveys is important, and working with the local people is important. This was done in collaboration with the Institute of Biodiversity in protected areas of the country and with one local university, and we are trying to training people to do this work, and we are developing many projects in this and other countries. So thank you. This river needs attention, and there are many rivers in these kind of regions that have never been monitored either with eDNA or otherwise, and there are huge opportunities that can be developed in these countries.
Thank you, Pedro. Also, the time for question will be lunchtime. Okay. We now have 3 shorter talks. We started a little late, so I would appreciate if most of you will stay in here for the last 3 talks. At first, it's about gut eDNA, and Anaïs Lacousière-Roussel, I hope I pronounced the surname correctly, will start to present her work on the centralized interactive guidance on optimal timing of eDNA.
That's not the one. The one.
Would be interesting, but
I was not prepped for that one. I can talk about that one.
Let's take another one. So that's That's only, I think, the last talk of the 3. I guess Katie is also here, but maybe no. That's the second one. But that's a good teaser, so you saw what interesting stuff will be coming, so stay in here. That would be a very general talk.
Hello? Apparently, there are only 2 talks, apparently.
Okay.
So I don't know where is the 3rd one.
Then I propose we start with Alison and search for the first talk. Apologies. Yeah, but for time reasons, let's start with the talk that was just up here from Alison Watts.
Hi. So, hopefully.
Yes, thanks a lot. So, building a standardized eDNA assessment and monitoring program in estuarine systems.
In 5 minutes or less. So, thank you. As with many of these projects, there are a ton of people who are working on this, way too many to put on the title slide, but we are a much larger group, and I'm merely representing them up here. I'm talking very briefly about a project with the National Estuarine Research Reserves in the United States. The NERS are a combination of NOAA, which is a federal agency, and they work with state agencies. So there are about 30 of these research sites around the country. They are all nestled within some state agencies. So like the one we're working with mostly in New Hampshire is working with, is in part of New Hampshire Fish and Game. The thing that's really interesting about them from an eDNA perspective is that they all do standardized monthly monitoring. They go out, they take samples, and specifically they take nutrient samples. When you take nutrient samples, you have to bring your sample back, you have to filter it within a very short period of time. So we've talked a lot about how one of the pinch points is collecting the samples and getting the people out there. Anywhere you have a nutrient sampling program, you've got people who are going into the field and have all the equipment to do this filtering. So it's a really easy add-on. What we're trying to do here is say, can we add eDNA into the standardized program in a way that is going to provide truly useful data? We're working with 10 pilot sites. So we're not working with the full 30 sites. We're just working with 10 sites for now. We have started. We've got about a year of data that we're really looking at now. We've got a little bit more, a couple more quarters to go. The sites, the staff at the sites collect the samples. They filter them there on site. They send them back to UNH for processing. Everything we're doing is pretty normal. We're doing triplicate samples, a variety of primers, looking at a couple of other primers coming up. And as we're starting to go through this data, what we're sorting through it, we're trying to understand what we have. We still have some time to change our course of action. We're getting pretty decent fish data. Most people are interested in fish, so we're pretty happy with the fish data we're getting. Some of the sites are a little more difficult. Looking at biodiversity and our 18S results, we're starting to be able to distinguish communities on different coasts. So look at whether or not the West Coast is noticeably different from the East Coast. I mean, we know the West Coast is different from the East Coast, but can we measure it in these particular samples? Ditto with North and South, and then eventually between different sites. Is that me, or is that, I'm not sure. But the place that is a bit of a pinch point, I'm just going to ad lib here, is that the sites are very different. So the sites are different. The quality of the samples, there we go. The quality of the samples is different, and that's deliberate. We wanted to pick sites that had a really range of site conditions and different types of biology, different types of access. But the part that's also challenging is that the end users, and we're trying to work very closely with the end users who are going to use this data, they have very different goals. So we come up with a really great fish data set, and the people on one site say, well, yeah, but we're really, really interested in copepods. Can you do copepods? So we can do copepods. But building a standardized program where we're doing everything the same, when we're starting with different materials, and we have people who want different things at the end, has proven, I think it's going to be a little bit challenging. And I think where we are headed is nailing down the stuff at the beginning. So the sampling and the extraction, because you can't go back and change that. But if you do that in a way that's the same at every site, and then you have a protocol pipeline that's, if not standardized, at least consistent across different sites, with some adjustment for the things that people are really interested in as you get to the end of that set of protocols. So starting very standardized, and then with some ability to adjust as you move down the pipeline. And then you can always go back and look at those samples again. And so that's, I think, where we were going with that. And our hope is that we can really develop a data set that does this inform environmental stewardship programs, which is in our title of our session here. Because if we're not meeting the user's goals, they're not going to use it, and it's not going to inform anybody, and it's just going to be another data set. And that's basically the talk. I will say that as we speak, I just said we're going to standardize our sample collection as we speak, right? There's a workshop in California where they're rolling out a whole bunch of really cool new automated eDNA samplers. And the thing we're always fighting, I think everyone in this room is talking about standardization, is cool new technologies. They wave it in front of you, and it's really hard to say no. Thank you.
Thanks, Alison. No, we will. I heard that only the old food will be served until 1:15, so we will wait until 1:15 when the new food comes. No. Next speaker is Katie Klimas. So she will speak about developing, so she's from USGS, and will speak about developing lab standard procedures and guidelines to improve uptake of eDNA methods. I learned a lot about the interesting results in Japan, and now the stage is yours.
Thank you.
It's here, and so that's your mic, and if you press
The green button? Okay. All right. Well, thank you, Florian, for that introduction. So I work for the U.S. Geological Survey, and this is an agency, a federal agency in the United States. And today I'm going to be talking about developing lab standards and procedures for the improved uptake of eDNA by resource managers. And I'm going to be talking about two large nation-scale projects that my collaborators and I are working on. And the collaborators are from the U.S. Geological Survey, but also a group from Canada. And so I'm going to be talking about the ReadyNet project, based in the United States, and the iTrack-DNA, which, if you were at the panel just right before this session, Valerie Langlois already kind of gave you a little brief introduction to that. Okay, so both of these programs their aim is to transition eDNA science from research into management. And one of the main sort of roadblocks that you come across is this, even though eDNA research and science has just increased exponentially, and there have been management applications of eDNA science, there's still a large hesitancy among natural resource managers and these stakeholders in the uptake of environmental DNA tools. Natural resource managers want to know, can eDNA tell me if a species is present, and how trustworthy is that data? So as a biologist at the U.S. Geological Survey, we work very closely and directly with many natural resource managers, trying to help improve the tools that they use. And one of these is of course, eDNA. But as I mentioned, there's a hesitancy. And some of the reasons for this limited uptake can be distilled to these 3 points here: poor communication about the state of the science, limited integration of decision makers in the actual development process, and the third one is the one I'm going to be focusing on: lack of trust and in the accuracy and reliability of this data. And I think this is a theme that has kind of come through the last omics panel and in these discussions. Are these results that we get, if I take a sample and send it to one lab, will I get the same results from another lab? And so this lack of trust can be in part due to a lack of standard methods, which leads to uncertainty or concern over the quality of these results. And so I'm going to first start by doing a brief introduction on ReadyNet. Hopefully you guys had a chance to see Devin Jones gave a poster on this project yesterday. But anyways, it is being led by Adam Sepulveda of the U.S. Geological Survey, and the idea is basically to develop an eDNA surveillance network across the United States. We're working closely with the Monterey Bay Aquarium Research Institute in developing autonomous eDNA sampling units to be spread across the United States, thereby increasing our input or throughput of eDNA samples that will be sent to a network of labs in the Department of Interior to be processed. And so it's a very large and multifaceted program, and the part that Maggie Hunter and I are focused on are the lab analysis standards. So making sure that these samples that come in, again, regardless of which lab they go to, we will get the same results. And I do need to emphasize that the lab protocols that we're focused on are qPCR and digital PCR, although we are interested in also implementing metabarcoding in the future. So for ReadyNet labs, for this facet, our goals are to develop standards and guidelines for these ReadyNet labs. We want to be able to assess repeatability and reproducibility, and also look at a cross-lab variation. And we'll be doing this through a series of inter-lab calibration studies, which will be actually are currently collaborating with iTrack on some of these inter-lab calibration studies to understand what variables could lead to variation among labs. We're also going to be developing proficiency testing metrics for both within and among lab variation, and finally develop a standards protocol or guideline for these specific labs. So while Maggie and I were working on ReadyNet on this program, we realized that both of our labs had also been asked by Karen Helbing to participate in this study, the iTrack DNA study, which is again being led by Karen Helbing, Valerie Langlois, and others. Similarly, they also want to be able to transition eDNA science from the research into application and management. Similar to ReadyNet, they're interested in understanding aquatic invasive species or pest invasions, but also looking at threatened and endangered detections through eDNA technology. And they also recognize that some of the current challenges are this lack or this hesitancy by managers of implementing eDNA tools, and that one of the ways to help push this forward is through standardization of these lab workflows. And so, as I had mentioned, iTrack and the ReadyNet programs are collaborating on a series of interlab calibration studies. And so there's an interlab calibration exercise in 4 phases. Phase 1 and 2 have already finished. But you'll see here we've got 17 labs from both the United States and Canada across various different sectors. Just really briefly, the idea is that we run multiple standard curves of a particular assay with a particular protocol and reagents. The limit of detection and assays are then measured for those assays across the lab to see what variation we see among the labs. I don't have time here to go into those results, but I'd be happy to discuss those later. I don't really know where I'm at with time, but that's my last slide. So, just to conclude, lack of method standardization contributes to this lack of implementation by managers. We're working collaboratively with another program, the iTrack program and ReadyNet, sort of this cross-country or cross-nation program collaboration to understand what leads to this variation. We are interested in developing guidelines, standards, proficiency testing metrics. And finally, we want to act as a sort of a demonstration or a pilot to managers to show that, yes, if we have standard protocols, standard lab workflows, that we can get reliable and reproducible results with eDNA. Thank you.
Thanks a lot, Katie. Do we have the last presentation available? Otherwise, we could have questions. Now the time's up. Thanks. Yes, that looks good. So, Anais, now it's your time. Anais from Canadian Government, Fisheries and Oceans Canada, and now she will talk about gut eDNA.
Yeah. Bonjour tout le monde, bienvenue à la province de Québec. Hello everyone, so I'm glad today to be here to present one of the new initiatives of Fisheries and Oceans Canada. It's called gut eDNA, so it's a guidance on optimal timing for eDNA. It's in collaboration with Parks Canada and Maine eDNA. Okay, so some species are releasing DNA in the water column randomly, but many of them are releasing DNA in a specific periods due to their phenology. So it can be reproduction, it can be biomass, or migration, for example. Then when it's released in the water you have then the difference between ecosystem due to the persistence of eDNA. So, for example, in the Arctic, we're expecting the detection window longer than other regions. So basically, the detection window, the optimal detection window, is the season, but also the duration of when you can go and collect samples. Okay, so last year we did an eDNA workshop, and it was quite alarming to see that how many eDNA groups do not consider the optimal timing periods when designing a new eDNA program. So that does mean that we are collecting a lot of false negative detection, but also that we're probably spending too many resources for detecting some species than others. So that's need to be integrated, but the problem is that even if we know that biological and physical factors are altering the detection window, it's quite hard to apply it and knowing how to adapt it to a specific regions or for a specific taxa within the literature review. So in the questions we asked, it was about 30% of the attendees mentioned that there are critical knowledge about how to adapt their sampling design. And about 40% said, well, we need advice to specific regions for specific taxa. So we did the first report of DFO. We combined 30 data sets, different projects all together. We were able to combine some of the projects because they were using the exact same field and lab methods. So total, it was 13 data sets, and we could provide when is the optimal timing for those data. That was the first comprehensive communication trying to explain how to adapt their sampling design. But it's about 300 pages long. So it's not a very efficient tool, and I actually wonder if you can really use our report to design your program, especially for biodiversity surveys. So here born the idea of developing something interactive where you can have access to the data, you can visualizing it. So we call that gut eDNA. And gut eDNA is basically a tool that you can use to visualizing your data, but also to have guidance on how to optimizing the sampling strategy. So the idea is that you can access to the data that are already published, but you can also import your data and just use it as a statistical tool. So there will be a data request window where you can have access to qPCR data or metabarcoding data. Then the user can select the taxonomy level that is interesting on, and then basically selecting a geographical region to know if there are data available and how it can be combined. So then there will be output of the observation window where you have the statistical value, so that describing the duration and the timing of the detection window, the optimal detection window. Then you have the variability between the fact that it can change between primers, regions, years, and having that idea of variability. Obviously, all of the script will be available on GitHub and freely available. And then you can visualizing the data. So here's just an example for white sharks. That was a data set collected in the Bay of Fundy, so we did it for a couple of years. And then we don't detect them early in the season, and we don't detect them late in the field season. So in November, we start to lose the sampling, the detection rate. That fit with the migration pattern. We know that in the Bay of Fundy, they're coming just for the summer, and then they're going back. So this year, we decided to just make all of our sampling efforts in the optimal timing window, so we could increase the spatial pattern. So it's just an example. And then you have visualization of the models and the variations within your data, what is significant. And then there's a data window, the last window. So that's basically aiming to provide access to minimal data information, having to know who is the data owner, and it's basically to the discretion to the data owner if they want to share the data or not publicly. And then there's a link that we hope to link with public databases. And then the gut eDNA number is basically when we consider that the observation can be combined together, or if they need to be not combined because they have different methods in general. Oops, next one. Here's another example for tunicates where we observed that the detection window was better in winter. For Didinum vexillum, which is one of the invaders, there was a significantly higher benefit of going in January. So that's basically a valuable information for a monitoring program that aims to monitor tunicates, the early detection when they are low abundance. Then there's another example for copepods. So that's for metabarcoding. So you can see the efficiency for the Acarcia, for 18S and CO1, and for Acarcia longirimus, where we didn't detect them with the 18S, we did have detected them successfully on that data set within CO1, and we have a successful detection window. So if you are interested in that species, well, you should go on that period. That would be more optimal. Cost-effective. We are also working on a prediction window that's still in development. So we are basically hoping that at some point when you select a region and it's not, we don't have a data observation yet, at least you can have some prediction created by the data that has been already imported and the variation that we have observed between regions. So obviously, more we are importing data within gut eDNA, the confidence will increase over time. Okay, so that's a collaboration with many eDNA and Parks Canada. So that's a data sharing agreement, a legal one, so that we have developed. And I think that's just a very good strategy. You know, if we are combining our data together, for example, in that case, the government of Canada, to have access to that data, then we can make sure that we are already having optimal sampling design with the data that have been collected from other regions, especially for the main that's a good example, where some species are expected to migrate in the water, so we can already be well prepped for having optimal sampling strategy. I would like to thank a lot of people because they are sharing data, and basically that's a very good collaborative project. I also would like to special thanks to Louis Bernatchez, who was my postdoc supervisor, and also my eDNA because they're really open, sharing ideas and not just data. So I think that's great to brainstorm all together, how to share data, how to report data, visualizing data, models, statistical models. So this is great. Communication, networking, and special thanks to the Inuit community because they're collecting hard-to-seq eDNA samples for us since more than 10 years now. So they have made a huge difference in our Canadian data sets. I just want to end with a special issue that we are hoping to launch very shortly. So if you are working on data management initiative, or how to share data, or application of how when we are combining data, I think that would be a good opportunity for you to share your data templates, your strategy in your lab, and things like that. So we just want to make sure that people are sharing their best strategy and having a good international synergy. Thank you very much, and I'm happy to take questions during lunchtime.
Thanks. Thanks, Anais. Yes, let me close the session with just a few remarks. I guess we all saw that there are many questions, and often I heard we still don't know what's here and there, but what we also saw is there are many solutions, and I think we will never be at the stage where we will know everything. But I think we convincingly saw that the technique is already there, especially for single species assays, but also for meta-barcoding work. It's now really that we find agreement on the minimum requirements and that we find this at the international scale. I would like to end the first part of this session, because tomorrow we will have the second part of the session, by first of all thanking all the speakers for their presentations. That was really excellent performance. So a quick applause for our speakers. And now I wish you a productive and hopefully tasty lunchtime, and I hope we see all of you tomorrow, and think about contributions to the special issue on standardization for meta-barcoding, metagenomics, same publisher as the Biodiversity Data Journal. Just get in contact with Merdad or myself.